Okay, wonderful. Well, it's a real pleasure to be here today virtually to share with you some of our research, um, really looking at trying to understand how psilocybin might be specifically therapeutically relevant to treating some of the symptoms of anorexia nervosa. And so I'll talk today in two parts, really. In the first part, I'll give a brief overview of sort of what the animal model of anorexia is. And in the second part, I'll talk about our recent studies using this model to understand some of the mechanisms of psilocybin that are relevant to anorexia nervosa. Now, of course, we use animal models not because we think they can recapitulate all of the complex psychosocial and biological interactions that occur in individuals with anorexia nervosa, but because they allow us to tease apart causal biological factors and to manipulate these in really precise ways. And so I'll start with some disclosures. First of all, none of the work that you see today was funded by any pharmaceutical or psychedelic companies. However, it is 7 a.m. on a Saturday morning in Melbourne, and it's 6 degrees outside. Additionally, I spent nearly 40 hours in transit yesterday because my direct flight home from Los Angeles was cancelled at the last minute. And I tested positive for COVID when I arrived home. So I can't really be held accountable for anything weird that might happen over the next 45 to 50 minutes. And I hope you'll bear with me because I have two cats that are not used to me being on Zoom at this time of the morning, and they may display this concern vocally. So my lab is dedicated to understanding the neurobiology of anorexia nervosa. It's the deadliest eating disorder. It has the highest mortality rate of any psychiatric disease. It kills more people than the annual road death toll in Australia, and has extremely high rates of suicide. And this is brought into sharper focus based on the fact that there are currently no approved medicinal treatments for anorexia nervosa. It's an intriguing problem to work on as a neuroscientist because it really represents a paradox for evolutionary biology. As humans, we've evolved to survive through biological adaptations that limit energy use during periods of food scarcity and that promote feasting when food is available. But the core features of anorexia nervosa are excessive exercise combined with minimal food intake, which is really at odds with the idea of homeostatic energy balance. And I think from a biological perspective, it provides a unique opportunity to understand and study interacting neural circuits. Restrictive eating is quite different from other psychiatric symptoms because eating is so essential to survival. So it must be the case that cognitive and emotional brain circuitries override these evolutionarily ancient hunger signals. And I think these interacting neural circuits might be the perfect target for new therapeutics. So we know from an expanding body of research using functional imaging studies in patients that are both currently ill or weight recovered from anorexia nervosa that multiple brain circuits are disrupted, largely related to cognitive and reward processing. And this is a complicated schematic of all of the different circuitries that have been identified. But an overall view that has been sort of highlighted in the literature of the last decade is that exaggerated prefrontal cortical activity combined with diminished ventral reward processing leads to an imbalance that enables disproportionate self-control over behaviour. And the consequences of this imbalance are, on the one hand, that people with anorexia nervosa are cognitively inflexible. They have a rigid focus on calories and exercise. They exhibit set shifting and reversal learning deficits on neurocognitive tasks, which really reflects an inability to modify deeply ingrained patterns of thought and behaviour, even in the face of starvation. On the other hand, diminished reward processing is evident in individuals with anorexia as a preference for delayed over immediate rewards in discounting tasks. Generalized anhedonia that relates to food and feeding, but also to other modalities, including social and tactile anhedonia. And habit-driven behaviour that is persistent, independent from the outcomes. But whether or not these neurobiological changes in patients are causes or consequences of anorexia are, of course, extremely difficult to determine without large-scale prospective studies where individuals are tracked from childhood before illness onset. So to understand how these neurobiological features might drive the development of anorexia, we turn to animal models, as I mentioned, in which brain circuitries can be perturbed and anorectic behaviour interrogated in detail. And so the model that we use in the lab is called activity-based anorexia, or ABA, and it's the most established and widely used animal model of anorexia nervosa that relies on giving animals, rats or mice, the combination of unlimited access to a running wheel paired with time-limited access to food. And it's important to note that food is only limited in terms of time and not quantity. And without the running wheel, animals will quickly learn to increase their food intake during this time window to maintain a healthy body weight. And so you can see this here in the blue line. Control rats that don't have a running wheel are able to maintain their body weight over a number of days. However, the addition of the running wheel elicits this precipitous body weight loss, reminiscent of uncontrolled weight loss in anorexia nervosa. This model's been around since the 1960s. And really one particular aspect of this model that is interesting is that, and sort of represents a failure to adapt to a change in the food availability, is that when food restriction starts, instead of reducing running wheel activity to compensate for the negative energy balance or the reduced food intake, ABA rats become markedly hyperactive, which you can see in running wheel activity in the purple dotted line. We've also recently, or not that recently, a few years ago now, shown that there exists subpopulations of rats that are exposed to these same experimental conditions that are either susceptible or resistant to weight loss. And so from this we can conclude two things. First of all, the weight loss or food restriction and hyperactivity is voluntary. And also it provides a unique opportunity to uncover factors that predispose individuals to pathological weight loss compared to those who are resistant. And there are a whole variety of other physiological and behavioural features of this model that mimic features of anorexia nervosa, including that adolescent females are most vulnerable, a loss of menstrual cycling, hypothermia, changes in hormonal regulation and metabolism, reward processing, mood and anxiety-like behaviour. But considering that a large driver of treatment failure and chronic relapse in anorexia nervosa are the cognitive disturbances, we've been most interested in whether ABA model also recapitulates some of these cognitive domains in order to understand more about why they develop, and by doing so, inform the development of novel treatments. And we think that this model is particularly useful for studying cognitive and reward-related deficits that drive anorexia nervosa, based on a pair of studies that we conducted using pathway-specific dreads or chemogenetics. And these were published a little while ago, so I won't describe them today, only to say that we could, if we increase the activity in this ventral reward pathway using the GQ or activating dread, or if we reduce and suppress activity in this prefrontal control pathway using the GI dread, we could re-establish the balance of reward and control in the ABA brain, in the rat brain, that is dysfunctional in human anorexia to prevent or rescue pathological weight loss. And we've also shown reciprocal effects of cognitive flexibility and activity-based anorexia using this reversal learning task, which is sort of depicted here in a kind of low resolution image, which I apologize about. Essentially, animals are trained to discriminate one image on the touchscreen from another by its delivery of a sucrose reward. And once they do this in the pairwise discrimination phase to sufficient accuracy, which is normally 80% accurate responses, the reward contingencies are reversed. And the animals have to adapt their responding in line with the new rules. And what we show is that ABA exposed but weight recovered rats perform worse, specifically on the reversal learning aspect of this task. And prior training with this task allows animals to resist weight loss in the activity-based anorexia model. And we've been able to do this using a really cool new apparatus that was developed for us by a company called Phenosys that uses RFID technology and a touchscreen chamber that's attached to a home cage via a series of gated tunnels where a sorting mechanism acts as the mechanical replacement to the experimenter. And so now we can run these touchscreen-based cognitive assays in a really high throughput way with group-housed individuals, so no social isolation, no stress associated with handling because we don't have to pick up the rats and put them in the chambers each day. And it really allows us to get a better understanding of the sort of ethologically relevant behaviours that drive cognition and how they might be related to the development of disordered feeding and exercise behaviour. Another cool device that we use in the lab is this home cage operant learning tool called the Feeding Experimentation Device, or FED3, which was developed by Lex Kravitz. And, you know, it's essentially a similar sort of vibe, except using nose poke responses instead of nose poking the touchscreen. And these devices can just be placed into the home cage of the animal to not disturb their normal behaviours. And these are particularly valuable for pairing with techniques to either manipulate or record from particular brain regions using fiber photometry or optogenetics. And so we can really precisely align behavioural responses with brain activity or manipulate brain activity to modify behaviour. And this is just some preliminary data using these devices in ABA rats. So after they've undergone exposure to the activity-based anorexia model and subsequently regained body weight to assess a particular aspect of cognition and learning called value-based decision-making. And it's really about how animals use goal-directed control to guide their behaviour. And to do this, we use a task called the outcome-specific devaluation task, in which animals are trained to associate one action, say the left nose poke, with one reward type, sucrose pellet, and the other action, the right nose poke, with a second reward type, which is a grain pellet. And then in the devaluation stage, one of the reward types is devalued by allowing animals to consume this reward type to satiety as much as they like before a choice test, which is conducted without any rewards delivered, but in which animals should bias their responding away from the action that is now devalued. And we show that after exposure to activity-based anorexia, responding is actually not impaired. In fact, ABA rats demonstrate this ballistic habitual responding with increased uncertainty of reward. So these are random ratios of reinforcement, random ratio schedules of reinforcement. RR10 means that you may get a reward once every 10 nose pokes on average. But what is impaired is their ability to discriminate between the valued and the now devalued action. And so this is the first evidence that prior exposure to this ABA model results in persistent compulsive behaviour. And consistent with the use of the utility of these fed devices, we're using a suite of tools, including fiber photometry, where we can record real-time activity using calcium sensors or real-time dopamine or serotonin release, and align those changes with specific aspects of behaviour during these types of tasks. And this is just a sort of snippet of what we can do, where we show that, as expected, activity in a particular region, sub-region of the dorsal striatum increases with training at a level of increased uncertainty. And so, we see the role of animal models as critical for informing the repurposing of existing neuromodulatory treatments like transcranial magnetic stimulation, to target specific symptoms of anorexia, including compulsive exercise or cognitive inflexibility or disrupted value-based decision-making. And all of these sort of circuit-based mechanistic studies, will help develop symptom-specific treatments that are maybe tailored to individual or clinical subtypes. And our goal with this work is to feed the knowledge that we gain from animal studies to clinical studies and translation into service providers and vice versa, in this sort of iterative way that I've represented in the figure here, in order to rapidly translate new therapies that actually work. But the rapidity or the rate of this translation is kind of slow. This work might have real outcomes for patients in 5 or maybe 10 years. But psychedelics are right here on the horizon. And what we need to understand is how they might be specifically therapeutically relevant for people with anorexia nervosa, and whether there are potential risks for different clinical populations. So, as you're probably mostly aware, there are 4 clinical trials currently underway in individuals with anorexia nervosa using psilocybin and assisted psychotherapy. And the first published small phase 1 open label feasibility study showed that it seemed to be well tolerated by people with anorexia nervosa. And our secondary measure of this study suggests that at least 4 out of the 10 participants also experienced long-lasting improvements in eating disorder symptoms after a single dose of psilocybin. And this is sort of a pretty standard response rate coming out of some of the other clinical trials. So, perhaps it's reasonable to question the need to study psilocybin in an animal model of anorexia nervosa. If it works, who cares how it works? And I think this image, which may have already been shown over the course of this conference, neatly encapsulates one of the main reasons that I think mechanistic studies of psychedelics in animal models are important for understanding the therapeutic outcomes, which is the intense media hype surrounding these compounds. Here at Newsweek, psilocybin is slated to be the next Prozac in the treatment of depression, but it's similarly unlikely to be a miracle cure, and the framing of it in such a way could actually result in worse outcomes for patients if they approach psychedelic treatments with an expectation of a miraculous outcome. And expectations matter in psychedelics trials because there's no placebo control for the intense subjective effects. And this could cloud interpretation of results from clinical studies, especially those that rely solely on self-report measures of efficacy. The other critical issue is that not all participants experience clinically significant improvements in psychedelic trials. And so we need to understand whether there is a biological basis for these response profiles in order to target therapies to individuals who are more likely to experience therapeutic outcomes. And so we know that some of the major neurobiological actions of psilocybin neatly align with the neurobiological features of anorexia nervosa, where individuals with anorexia nervosa show exaggerated prefrontal cortical activity, disrupted serotonergic and dopamine signaling, and impaired cognitive flexibility. But how exactly psilocybin creates these changes, and whether we could tailor the application of psychedelic treatment to people most likely to respond in anorexia nervosa, requires direct interrogation of these mechanisms of action in the brain. And so, considering that weight loss in activity-based anorexia occurs via a failure to adapt behaviour to a change in food availability, we hypothesised that a single dose of psilocybin after the normal period of wheel training and 24 hours prior to the onset of the ABA conditions could improve this ability to adapt and prevent weight loss. So no drug was on board when animals had restricted food access. The administration occurred 24 hours prior. And what we show is that the body weight trajectories of individual animals over the 10 days of food restriction and wheel access, or ABA conditions, demonstrate a significant curtailment of body weight loss in the psilocybin-treated animals. We showed that the initial weight loss was curtailed by psilocybin, and the severity of weight loss was also reduced. I think it's also really neat and interesting that not all animals responded to psilocybin with improvements in weight maintenance, which was sort of similar to the response rates in clinical trials. So we're working on trying to understand what differentiates these responders from non-responders. And it doesn't appear to be any aspect of the activity-based anorexia model itself. These are all psilocybin-treated animals that either responded with positive body weight outcomes or not, and they're just better at they just do better at maintaining healthy behaviour across all aspects of the paradigm. But so we also evaluated how psilocybin directly influences cognitive flexibility using a reversal learning task similar to that I described with the touchscreens, only this time using the FED3 devices. And when psilocybin is administered following training and the day before the reversal of the reward contingencies, you can see that rats treated with psilocybin can learn this new rule much more quickly than saline-treated controls. And so this is accuracy of responding to the newly rewarded side. And in the first day, we see a significant increase in performance after psilocybin treatment. Psilocybin also increased the proportion of animals able to learn the task within this first day and shifted the rate at which animals are able to adapt their responding. So they needed less trials or less pokes, less reinforcement to reach this performance criterion of 80% accurate. Interestingly, the effect of psilocybin on appetitive learning wasn't generalizable to other tasks using these strategies. We saw no differences between psilocybin and saline-treated animals on a task of motivated or effortful responding called a progressive ratio task. They were not able to better suppress responding on an extinction task, and both of these behavioural features could potentially explain the improvement in cognitive flexibility. You know, maybe they're just more motivated to perform, or maybe they're better able to suppress their previously learned response. That doesn't seem to be the case. And response vigor under uncertainty, so how vigorous they're likely to press for reward, was unchanged, as was suppression of this sort of variable ratio training, unchanged by psilocybin. So we're interested to know whether this specific improvement in flexible learning and an improved ability to adapt to a change in rules was mediated by the actions of psilocybin at the 2 serotonin receptor subtypes that have shown abnormal binding profiles in people with anorexia nervosa. These are the 5-HT1A and 2A receptor subtypes. And to do this, we administered selective antagonist compounds 30 minutes prior to administration of psilocybin or saline, with the view that these antagonist compounds would block the actions of psilocybin, specifically at one or the other receptor subtype, and repeated the reversal learning task in separate cohorts of animals. And we show that antagonism of the 5-HT1A receptor subtype, but not the 2A receptor subtype, significantly blocked the psilocybin-induced improvements in performance. And you can see that here in the accuracy on this first day of reversal, with the 1A antagonist plus psilocybin in the light blue color. That performed very low accuracy. Very few actually reached performance criteria. And this is sort of another measure of accuracy at the end of the session. Interestingly, this was not associated with a perseverative phenotype. So you would expect if they were perseverating on the previously rewarded side, that they would make more non-target pokes per session. They don't seem to do that. But it just seems to be that they, with the 1A antagonist on board, they were unable to switch to the new target side. And they also took longer to make the first correct poke. But the 5-HT2A receptor is also involved in the effects of psilocybin on reversal learning, because when we compare performance of animals treated with the 5-HT2A antagonist itself, compared to co-administration of the 2A antagonist and psilocybin, we see a huge impairment elicited by the antagonist itself, an effect that was partially restored by psilocybin, at least in terms of response accuracy. So both receptor subtypes are involved in the effects of psilocybin on learning, but does psilocybin itself alter the expression of these receptors in the brain? And the place in the brain that we looked was the prefrontal cortex, obviously a known site of psilocybin action and critical for cognitive function. And we used RNA-scope, which is a fluorescent in situ hybridization technique, to quantify the effects of psilocybin on the abundance of these receptor transcripts, so the individual mRNA transcripts of the receptors, to look at the relative balance of one receptor or the other on specific cells in the prefrontal cortex. And hopefully you can see on this micrograph here, each of the blue blobs is just a DAPI stained cell. And each of the green dots is a single 5-HT1A mRNA transcript, and each of the red dots is a 5-HT2A mRNA transcript. And what we did was a time course study where we administered psilocybin and collected brains from rats at 6, 12, and 24 hours post-administration. We saw no effect on the number of cells that express, no effect of psilocybin on the number of cells that express both receptors. So these are double labeled cells. So I haven't shown them on this graph. But what we did see was that psilocybin elicits this transient increase in cells expressing only the 5-HT1A and a co-occurring or concomitant decrease in the cells that exclusively express the 2A receptor. And this effect was more prominent in the infralimbic than the prelimbic subregion of the prefrontal cortex, medial prefrontal cortex. And it was also specific to layer 5. And so these are the number of transcripts measured in microns from the midline of layer 1. And we can see that the output layer, layer 5 of the prefrontal cortex, is where the effect of psilocybin seems to be most significant. And so the net outcome of these changes on cortical activity is presumably suppression of cortical output, because binding to the 5-HT1A receptor is generally inhibitory and reduces neuronal firing, whereas binding to the 5-HT2A receptor is excitatory and increases neuronal firing. So what psilocybin is doing, we believe, is increasing the potential for inhibitory output and reducing the potential for excitatory output from the prefrontal cortex. We, of course, wanted to know whether these effects also happen in rats exposed to the activity-based anorexia paradigm to the same extent as in healthy rats. But for practical reasons, we collected the brains of these ABA rats at 6 hours after administration of saline or psilocybin, even though the effects that we saw in the time course study were most significant at 12 hours, because we didn't want them to spend too long being underweight after they reached the body weight criterion that we deemed determinant of susceptible in this study. And by and large, the effects of psilocybin on the number of exclusively 1A or 2A expressing cells was recapitulated in the ABA rat brain. And this finding was really important because there's been a lot of discussion in the clinical space about whether or not psilocybin would even be effective in a human brain that already has altered serotonergic receptor expression or serotonin receptor function. And when we looked at not at the level of the cell, but at the level of the RNA transcript, so the single dot, we show a significant reduction in the abundance of these 5-HT2A transcripts on each cell that was elicited by psilocybin. And this was specific to ABA rats. So we think what this means is that the propensity for psilocybin to cause neuronal suppression is enhanced in the situation of anorexia, at least at this transcriptional level in the prefrontal cortex, but that might be therapeutically relevant for people with anorexia nervosa. And so to summarize this data set, it seems that both receptor subtypes play a role in early reversal learning, and that 5-HT1A function may be required for the effects of psilocybin on improved cognitive flexibility. We also show the transcriptional changes in these receptors are transient, resolve to baseline levels within 24 hours, but produce net suppression of the output from the prefrontal cortex, and that this potential for suppression is further enhanced in ABA rats. However, it's important to recognize that these receptor subtypes are co-expressed on the same neurons, have pre- and post-synaptic locations, and anatomically distinct expression on both inhibitory and excitatory cells, none of which we could determine from the type of assay that we ran using RNAscope. So exactly how their activation by psilocybin impacts on neuronal excitability and downstream molecular signaling isn't clear from this study. So what we need to do now to understand how specific changes in the balance of expression of oppositely acting 5-HT receptor subtypes is to identify the phenotype of the cells on which these changes occur. So we know that a large proportion of pyramidal glutamatergic cells in the rat prefrontal cortex co-express both 5-HT1A and 5-HT2A receptors. And we know that there are two populations of interneurons, inhibitory GABAergic cells, that express one or the other receptor cell type. So we have these fast spiking parvalbumin positive cells that express the 2A receptors, and we have these low threshold spiking somatostatin expressing cells that express the 1A receptor. And so teasing apart the effects of psilocybin on these cell types is one of our next major jobs. So just as a little bit of an aside to use some of the time that I have left, I wanted to share some of my thoughts on how we can better use animal models to both study the neurobiology of human pathologies, but also the actions of psychedelics. And this sort of has been percolating in my mind for a little while, based on the fact that this activity-based anorexia model has been around for more than 50 years. I sort of alluded to that at the start. And so after 50 years, more than 50 years of using this model, why don't we have any concrete answers about how anorexia nervosa is underpinned by changes in neurobiology, and why do we still not have any approved medicinal treatments for this disorder. So I won't go into all of the details of my musings on this topic, but there's a code there that links to an opinion piece that I wrote earlier this year or at the end of last year. If you're interested to understand how I think we can take better advantage of these types of animal models. But I will just sort of, make three main recommendations for improving the translational capacity of any animal model of a human pathology, but with particular, emphasis on this activity-based anorexia model. And this is a quote I like to use a lot by a statistician, that all models are wrong, but some are useful. And I think that's important to recognize, particularly in the psychedelics space, because none of the animal models that we use to study the mechanisms of psychedelics perfectly recapitulate human psychopathologies. But perhaps refining and thinking deeply about the design and the use of the models that are employed will improve our ability to both understand how psychedelics work and find better treatments for psychiatric illness. The second sort of take-home message, I suppose, is that collaboration and consensus is critical. So, at least for people working with this activity-based anorexia model, and I'm sure it's the same for a range of other different animal models of human pathologies, that different parameters are used across labs, different feeding and habituation, different running wheel times, different ages of animals. And this really makes it hard to generalize findings across different laboratory settings. And so my suggestions are both to publish negative results. It's really important for the field to understand what doesn't work, and to standardize practice and parameters for understanding the same phenotypes. Finally, we need to embrace new technologies. New neuroscience technology has been our friend in the lab over the last several years. And I think, using this creatively will allow us to bridge disciplines and incorporate neuroscience with genetics, psychology, metabolism, immunology, a whole range of fields for a more comprehensive understanding of both the biological drivers of psychiatric conditions and the mechanisms of actions of psychedelics. And so I'll briefly share one example of how we are rethinking the utility of the ABA model and how this may shed some light on why some individuals respond with positive outcomes after psilocybin treatment and others don't. And so I'll take you back to this slide where you can see the trajectories of body weight loss in individual animals exposed to the activity-based anorexia paradigm. But for now, forget about the psilocybin part, focus on the weight trajectories and focus on the substantial variability, even among controls, in the rate or the trajectory of weight loss. So we have this kind of arbitrary ethics committee directed cutoff for susceptibility to ABA when animals lose or drop beyond 80% of their baseline body weight. They either go back onto full food or are removed from the experiment. But our observations have been that some rats at this body weight loss criterion seem completely fine, whereas others look obviously unwell. And it's these types of observations in the lab that, need to be thought about critically and deeply to drive innovation in these types of endeavors. And so we wondered whether there was a basis for these differences in weight loss trajectories, and whether maybe there were more than just two subgroups of animals. Maybe there was an intermediate phenotype that may be biologically distinct to very susceptible and very resistant animals. And so we took a couple of cohorts of rats and used a viral trap approach where we inject a retrograde transporting Cre into the nucleus accumbens and a Cre-dependent trap virus into the prefrontal cortex. in this circuit that we'd previously established to be involved in the development of weight loss in the ABA model. And we selected, 4 resistant rats, 4 susceptible rats that showed extremely rapid weight loss, and 4 intermediate rats that meet the criteria for susceptibility, but at a much slower rate. And we sequenced these projection neurons and compared the 2 susceptible subgroups to animals that are resistant to weight loss. And to our surprise, there was 0 differential gene expression overlap between the rapid weight loss losers and the intermediate weight losers, which suggests that these animals are different to resistant rats for entirely different reasons, at least within this neural circuit. And there are some interesting genes that come out as differentially expressed in the 2 subgroups of weight loss susceptible rats. And we're really just in the process of diving into the analysis of this now to make some sense of how these groups might differ biologically, whether we can use this to better model the types of cognitions and behaviours that are present in individuals with anorexia nervosa, and whether these differences might explain the differences in response profiles to psilocybin. More broadly, the lab is focused on understanding more about where and how the serotonergic effects of psychedelics interact with other neurochemical systems. So we have on the diagram here the dopamine system, but we're also interested in other neuromodulatory actions of psychedelics. Our view is to understand whether the interactions between different neurochemical systems can be harnessed to develop new drugs with specific modes of action, and in doing so learn more about the fundamental drivers of decision making and cognitive control. And so with that, I would like to thank the lab. Here we are. Everyone plays a vital role in the outcomes that we produce experimentally and conceptually. I want to thank collaborators that have contributed to this and other work in the lab and that are contributing to ongoing work. USONA for providing the psilocybin that we've used in these studies, and the funding that we received from the Australian Eating Disorders Research and Translation Centre, the Australian National Health and Medical Research Council, and some foundation philanthropic funding from Rebecca Cooper and the British Society for Neuroendocrinology. And hopefully we have time for questions. I'm not entirely sure, but I will end my share and see what we have going.
Yes, we certainly have time for questions. First of all, I just wanted to thank you so much for being here so early on a Saturday, and then also after all that traveling and being ill and everything. So you're certainly an inspiration to all the students in the audience that you're able to deliver such a wonderful talk after all that. So thank you so much. We do have a few questions. I'll start with this one. Do you have any idea as to the mechanism behind why rats become hyperactive upon food restriction?
Yeah, I mean, a lot of people are working on the reason why. Part of it, I think, is dopamine related. So we've been looking at the changes in dopamine release in the dorsal striatum. A group in the Netherlands also works on dopamine and the interactions between dopamine and leptin. So leptin is a hormone that's produced by fat cells. And so as leptin levels decrease with declining fat mass in these animals, that triggers a increase in dopamine that may underlie the hyperactivity. But I guess the bottom line is we still don't know. It's a super interesting phenotype, paradoxical phenotype that a lot of people are trying to work on what drives it.
I see. And kind of related to that is, so as we know, anorexia in humans typically affects females more than males. So do you see any sex differences in the ABA mice that are maybe more susceptible to the model or less susceptible?
Yeah, This is also super interesting and inexplicable in some ways. We have run a few, so we use female animals exclusively for most of these studies. We have run male animals through the same paradigm at different ages to try to see whether or not we can get the same phenotype in males in wild type Sprague-Dawley rats. And what happens with males is that they just don't engage in that food restriction evoked hyperactivity. So they'll develop a baseline level of running wheel activity that's sort of, individually variable, but that's quite stable. And that it continues to be that stable when food restriction starts. So in our hands, male rats actually don't develop the activity-based anorexia phenotype. Some people have shown that male mice, black 6 mice, do develop the phenotype, which may have something to do with the inbred nature of the C57 black 6, where males and females may be a bit more genetically similar.
Wow, that's fascinating. And to me, that kind of almost lends more credibility to the model that it's, more of can serve in more of a translational capacity, just those sex differences.
Mm.
All right, here's another question. Do you know if any other serotonin receptor agonists, such as LSD, also have the positive therapeutic effects on anorexia that psilocybin does?
No, we don't know yet. So, our focus in the lab has been on psilocybin mainly because it's been the focus of the clinical trials. As far as I'm aware, there hasn't been any reports of LSD in people with anorexia. There was one, ayahuasca study in which, people didn't necessarily disclose eating disorder symptoms. They weren't necessarily clinically diagnosed, but several reported improvements in sort of body image and eating behaviours after an experience with ayahuasca.
Interesting. All right. Chantal asks, maybe I missed this, but if anorexics have less body fat, then why are they not having as much suppression of satiety from decreased leptin?
Sorry, if they have less body fat.
Yeah, they have less body fat. And Chantal, feel free to, if you would like to come off mute, or just raise your hand. So if you would like to clarify, if they have less body fat, why are they not having as much suppression of satiety from decreased leptin?
Yeah, I mean, they don't feel hungry.
Okay, here she is. Let me.
In fact, often people with anorexia report that feeling of hunger as euphoric, not uncomfortable, or a feeling that promotes motivated feeding. So
Okay.
the interaction between those sort of hunger signals and cognition, it is sort of what I was alluding to at the start as being a really interesting, complex entanglement that needs to be uncovered, because the very basis of hunger and satiety is disrupted in people with anorexia.
I see. I joined a bit late, so I didn't realize that was already covered. So you're basically saying they're not feeling full because they have less leptin, but they actually find that feeling of not feeling full euphoric, and that's because it's like dopaminergic in a certain way. And like, one thing I've heard is that LSD is one of the more dopaminergic psychedelics. Not sure exactly how accurate that is. I'm wondering if based on the previous question, that would contraindicate the use of that for people who have anorexia, or maybe like.
No,
or
I don't think so necessarily. I mean, I think there are, equal, if not more, evidence to suggest dopaminergic dysfunction in individuals with anorexia nervosa as serotonergic ones. I think, psilocybin has probably been selected over other psychedelics for a lot of these clinical trials, because of its time. The time of effects is a bit more clinically amenable than a very rapid acting or a very long acting psychedelic. But yeah, I don't see the dopaminergic actions of LSD necessarily precluding its therapeutic relevance for anorexia.
Got it. And do you think for people who are having trouble, I mean, this is obviously, big stretch, but is there any kind of model for people who may, from like chronic illnesses, have like low appetite, like have benefit from psychedelics, or is that not really been studied?
I don't think psychedelics necessarily change appetite per se.
Okay.
I think they change these cognitions that become distorted as that either drive persistent restricted food intake or that are a consequence of. Psychedelics acutely at least are not appetite inducing, they're appetite suppressing if
Right.
anything acutely. So
Right.
yeah, I guess more
It's more,
research is needed across the board,
yeah,
but
I
I
see. So
think
it's more
rather than a
mental
specific
behavioral.
effect on hunger or satiety, it's really
Yeah.
it's treating this cognitive and reward
Got
related
it.
disturbances instead.
Got it. Okay, that's good to know. All right, thank you.
Chantal.
Oh,
Sorry. Did you have another question?
no, I didn't have any more immediate questions. I wanted to make sure other people's questions could be answered.
Okay, thank you. All right, I think we have time for one more. So, have you studied different doses of psilocybin? And are hallucinations necessary for the effects that psilocybin has on anorexia?
We have done some pilot studies with different doses. So we've looked at 0.3 milligrams per kilo and 3 milligrams per kilo, neither of which seem to have a specific effect on body weight in the model. So the high dose, just really knocked them around, and the low dose didn't seem to have any effect. The hallucinogenic effects, I suppose, are the 2A mediated effects. We haven't done the antagonist studies in the activity-based anorexia model. We did all of the antagonist pre-administration in the reversal learning paradigm, and the results suggest that that if you block the subjective or hallucinatory aspect of psilocybin effects, as in if you can say that it's binding to the serotonin 2A receptor, the animals were still better able to perform the cognitive tasks than control animals. So I guess in that way, the hallucinogenic effects are not required for the not necessarily required for the improvements in cognitive performance.
Okay, wow, that's fascinating. All right, well, thank you so much for being with us today. If any students in the audience would like to learn more or get in touch with you, who do you recommend they look up or follow? Or
absolutely, just look up our lab. Look up, Google my name, and you'll find something. And the cat's here.
you.
Come on, mate.
She was polite and stayed out for most of the time.
All right. Yeah, great. Thanks very much.
All right. Thank you so much.